My name is Darrell Falk. I'm an emeritus professor here at Point Loma Nazarene University of Biology, and it's my privilege to introduce Dr. Hood. And just a few words then by way of introduction. It's only 60 years since the discovery of the structure of DNA, and along with Darwin's discovery of natural selection and evolution, there is no other single breakthrough that has more thoroughly reshaped the discipline of biology than the elucidation of the DNA structure, of course. In shaping the direction that DNA's footprints have left on biology, there are several towering figures who stand out. By any estimation, Dr. Leroy Hood is at the very forefront of innovators and thinkers who have played key roles actually, in marking the pathway for DNA. Without the technology to take the ramifications of DNA from the lab to the place where each person lives their lives, knowledge about DNA would be far too esoterical. It would be locked inside the minds of scholars and the labs of research universities. No single person, I'm not exaggerating here, no single person in the last 60 years has done more to catalyze the life-changing benefits to society than today's speaker. Dr. Hood has played the key role in inventing and bringing to the world the automated protein sequencer and synthesizer, and the remarkable instrumentation for DNA sequencing, DNA synthesizing, and creating DNA arrays that has made the current genome revolution possible. You know, it was against all odds, actually, that the Genome Project began. The majority of leading molecular biologists were opposed to it when the idea was first presented in 1985. And over those next 4 or 5 years, it continued to be something that was touted by a small number, but the majority actually were opposed to it, 90% by most estimates. But Dr. Hood not only provided the instruments to make it possible, but the enthusiastic political wherewithal to see it become a reality. We're benefiting it from today. Dr. Hood hails from Missoula and Shelby, Montana, where he was a quarterback of his high school football team. His lifelong love for the outdoors was fostered there. His diversions from the intensity of high-powered science include mountaineering, hiking, running, photography. As a college student, he played football on the Caltech team. And since their home games, there were 4 a year, were played in Pasadena's Rose Bowl Stadium, he has played in the Rose Bowl more times than any living scientist. He has published over 760 peer-reviewed articles, has 37 patents, and has founded 15 companies. He has won numerous awards, some of which you saw, including the Lasker Prize for his seminal work on the basis of antibody diversity, and the Kyoto Prize for his development of automated instrumentation, and the National Medal of Science, along with many, many other equally prestigious awards. He earned both a bachelor's and a PhD from Caltech and an MD from Johns Hopkins. He began his career as a professor at Caltech and went on to head the prestigious Division of Biology at Caltech for many years, and also headed its Cancer Center and Caltech's NSF Science and Technology Center for Molecular Biotechnology. In 1992, through the support of Bill Gates, he moved to Seattle and the University of Washington, where he founded the highly innovative and interdisciplinary Department of Molecular Biotechnology in the medical school, and where he also held and still holds a number of appointments. Then, in the year 2000, he founded the Institute for Systems Biology, of which he is currently the president. It is truly an incomparable honor to welcome Dr. Leroy Hood as our plenary speaker as he talks to us on the topic, Systems Medicine and Proactive P4 Medicine, a Revolution in Healthcare. Dr. Hood.
Well, it's a pleasure to be with you today, and I'll give you a personal journey that started in my earliest days when I was a molecular immunologist and has evolved through many of the things that you've heard about. Up until today, we think we can transform healthcare with the approach that I'm going to be talking about at the very end of my lecture. I will say that biology is really about complexity. Medicine is really about complexity. And the complexity of these endeavors means we aren't going to solve the biggest and the most challenging problems by studying one gene or one protein at a time. That is, we have to take a much more sophisticated kind of approach. I remember in high school first thinking seriously about complexity when, as a senior, I helped teach sophomore biologists, and I did it by using Scientific American articles. And an article that really stayed with me was an article on the structure of DNA. And I was enormously impressed with the apparently simple elegance of the DNA molecule, but I was also utterly perplexed that such a simple molecule could lead to such complex organisms as ourselves. And in fact, when I went to Caltech in 1970 and started thinking about what I wanted to do in my professional career, this whole idea of complexity really emerged again, because I knew by that time that I really wanted to do medical research in one way or another. And I think what is really interesting is the complexity is very much like this parable of the elephant and the 6 blind men. Each of the blind men feel a different part of the elephant, and they confidently report that the elephant is a spear, or it's a rope, or it's a fan, when in fact the elephant is all of those things and much more. And of course, in dealing with the complexity of the elephant, we lacked at that time a conceptual framework for thinking about complexity, and that has subsequently come to be systems biology. We'll talk about that. But as well, we lacked the technologies and the strategies for obtaining information about the elephant that could be integrated together to give a true picture. And in thinking about technologies and strategies, I remember reading in 1973 this book on the structure of scientific revolution by Thomas Kuhn, and it was a book about paradigm changes in physics. So a paradigm change is a thought that transforms our understanding of some aspect of science. And what Kuhn said about paradigm changes were 2 things. One, they were really hard to effect because true paradigm changes required dramatic alterations in how one thought about particular problems. And they were equally hard to attain because most scientists are incredibly conservative, and they're very reluctant to change the ways that they learned in their youth. And I came to understand in detail exactly what he meant by this. But over the last 50 years or so of my career, I had the good fortune to be involved in a series of 5 paradigm changes that collectively did let us deal with complexity on the one hand, but on the other hand, led us in a steady progression to this new kind of medicine that we're talking about. So the first was the one that you've heard about already. We developed 6 instruments that allowed us to read and write DNA, and the real contribution those instruments made, apart from a framework for how molecular biology was done, is that it opened up high throughput biology, the ability to make many rapid measurements, and it opened up the era of big data and its analytics, and we'll talk in a great detail more about that. One of these instruments, as you've heard was the automated DNA sequencer. Why, this doesn't seem to want to advance very well. Was the automated DNA sequencer, and that got me in 1985 invited to the first meeting ever held on the Human Genome Project. And it was an interesting meeting. Robert Sinsheimer, the chancellor there, had thought seriously about starting an institute to sequence the human genome, and as a consequence, he invited 12 scientists that knew in various ways about genetics and human biology and the genome to come and discuss this for 2 days. And we came to two conclusions. One, it would really be difficult, but was going to be technically possible. And two, we were split 6-6, and those against it were really fanatically against it. And as I went out into the community over the next 5 years to advocate for the Genome Project, as you heard, initially 90% of the people were opposed for really traditional doctrinaire grounds. But in the end, it was this conservative, I don't want to change the way I think kind of thing. But the Genome Project did happen. And of course, its major contribution was, for the first time, we had a complete parts list of all the genes and by inference the proteins, so we could think in a systems way about how to approach the biology of the human and the other organisms whose genomes were available. As a consequence of developing the automated sequencer, boy, I wonder, is there another clicker that can work reliably? Because this is going to be hard if we have to go back and forth. We had to put together a team that included a chemist, an engineer, a computer scientist, and a molecular biologist. And I realized at that time, departments of biology ought to be cross-disciplinary in nature, because you wanted leading-edge biology to drive technology, and that in turn to push the development of the analytic tools you needed to handle the information. And if you could put those things together in a reasonable fashion, then you could really transform biology. So as you heard, Bill Gates made it possible to start the first such department at the University of Washington School of Medicine. And it was a spectacular department. It developed the first two critical technologies in the emerging field of proteomics. It developed the key software for the Human Genome Project. We developed the inkjet technology for DNA array synthesis at that time, and on and on. So it was really quite a transformational department. But what happened was the dean that invited me to the university had guaranteed that I could have another floor to start systems biology after 4 years. And that dean, unfortunately, at that time, died in an avalanche in the Himalayas. And the next dean had a very different point of view about how to do things. And so it was after trying 2 or 3 years, I decided to resign and start the Institute for Systems Biology, first institute that was focused on this global, holistic view. We'll talk about what it is in just a few moments. But it almost immediately began thinking about systems applications to disease. And of course, that led to the concepts then of systems medicine and proactive P4 medicine. And that's mostly what we'll end up talking about. But let me tell you a little bit about my view of systems biology, and then we'll go on to systems medicine and P4 medicine. So what is it? Well, I would say first, it's a way of thinking about biology, a way of thinking about systems, and that is to view it in as holistic a manner as possible. It's also being able to have a parts list so you can look at all the individual components in the system. And of course, not only define them and come to understand what they do in singular terms, but how do all these different components interact with one another. And of course, what's really important is how do they interact dynamically, both changing what they do in time and in space. And what's really critical about systems biology is that you have the ability to perturb the system so you can test hypotheses and come to understand mechanisms. What lots and lots of big data experiments do today is they make really striking correlations. The trouble with correlations is you can explain them many different ways. There is only one proper mechanism, and perturbations are necessary to get at that mechanism. And of course, if you put these all together, you can understand both the function of biological systems and the basic mechanism. So that's the kind of global holistic view of what systems biology is. And of course, systems medicine is exactly that, only it's applied to disease rather than to biology. So I'd say there are two fundamental features of systems medicine. One is the idea that in the very near future, each of you will be surrounded by a virtual cloud of billions of data points of many different types, ranging from molecular and cellular data all the way up to organ and even social network type of data. What we'll be able to do with these data are integrate them and model them for each individual so we can identify the actionable possibilities that will let us optimize your wellness and minimize your disease. And I'm going to talk a lot about what I mean by that at the end. Now, what I will say about big data that's really important to understand is there are lots of computer scientists that are utterly fascinated with machine learning and how give me a lot of data and give me machine learning and I can learn everything about biology. And I'll say unequivocally that is utter nonsense. And it's utter nonsense because in big data there are enormous signal to noise issues. And most of the noise is biological noise of irrelevant systems you don't care about. The only way to get rid of that noise is to have a deep domain expertise for biology. So whatever you're thinking about doing in biology, learn the fundamentals of biology really well. Computational tools are, I'm going to argue, are essential as well. Now, one of the things we can do with those data clouds is we can extract from them a whole variety of biological networks, which are the informational conduits for mediating development, physiologic responses, aging, and if those networks become disease perturbed, they regulate disease. If you can capture the nature of the perturbations in those networks, you can come to understand in a deep way mechanisms of disease, and you can create tools for early diagnostics and even therapeutics. And we'll talk about that more in just a few minutes. Now, about 2006, we were really in this unusual position of having clearly defined systems medicine and P4 medicine. We'll talk a little bit more about that later, obviously. But the question that I was really struck with is how can we bring these startling new approaches to thinking about disease to the healthcare system? And that was when I really had a fortunate experience. I met the Minister of Finance of the state of Luxembourg, and he at the time was thinking about diversifying Luxembourg's economy away from a 90% dependence on financial services to bring in healthcare and biotech. So he said, would you be willing to write a proposal that would enable us to do that? So we did, and we wrote a proposal where we did a lot of things for Luxembourg, which I'm not going to talk about. But what Luxembourg did for us was give us $100 million over 10 years to invent the strategies and technologies of systems and of P4 medicine. And what was unusual about this money is it was, one, totally flexible, so you could take on high-risk, high-gain projects that never would have been funded by NIH. And number two, you had sufficient money to explore these challenging approaches in enormous detail. And what that gave us was we developed 10 technologies and our strategies, and I'll show you a few of these in just a few moments. But they placed us at this marvelous tipping point where it was clear we really could transform healthcare in major ways. And so the question was how to execute on all of these possibilities. And it was in 2013 when I first made the proposal that the way to do that is to create a large pilot program where we would do a longitudinal study on 100,000 well individuals and generate for each of them the very deep data cloud that I talked about earlier in systems medicine. And we'll talk in more detail about that later. And of course, Obama's 2015 initiative on precision medicine, though poorly defined at this point in time, could conceivably be a source of funding for this endeavor. But again, in 2013 and 2014, we were, could you advance it again? Great. At this really marvelous point where we developed all these different kinds of technologies here, and I'm not going to go through them except to say there is a third generation DNA sequencing technology coming. It'll be here in 5 to 8 years. It will allow us to do single molecule sequencing. We'll be able to do 100 kb sequence reads. We'll be able to identify all of the 16 epigenetic marks present on DNA. We'll be able to do RNA sequencing every bit as easily as we can do DNA sequencing. And we'll be able to multiplex this strategy to the point where I think human genome will be $100 or less. So it's a simple test, and you'll all have your genomes done. You'll probably have, many of you will have your genomes done before then, presumably. But there are other techniques, and I don't really have the time to go into them. But the systems-driven strategies are really equally important because it's taking a systems approach again to complexity. So how do we get disease dynamics? We develop family genome sequencing to identify disease genes, and it's an incredibly powerful technique for doing that. And we've really made blood a window into distinguishing health from disease, and we've done a whole series of other things I'm not going to talk about, but let me talk about each of the first three of those things. The first real disease we studied in detail was a prion-induced neurodegeneration in mice, where we knew when T equals 0 was in the beginning of the disease. And what we were able to demonstrate is, one, that there were 10 major networks that became disease-perturbed. Number two, that those networks became disease-perturbed in an interesting sequential fashion, starting with the most unique network at the very beginning. And number three, if you look collectively at the dynamics of those networks, and this was all done by gene expression patterns, you can explain virtually every aspect of the pathophysiology of this disease. And of course, what was really interesting is these dynamics revealed that the changes toward neurodegeneration in the onset of this disease manifest themselves more than 6 weeks before any of the clinical signs were initiated. So we can identify very, very early tipping points if you study the brain. But what we went on to, and I'm not going to show you this data, is we studied the blood in these mice, and we could follow the sequential disease perturbation of all of those different networks and pick up the changes at about the same time we could from studying the brain. So we had the ability to look at tipping points and transitions and going from wellness to disease. So keep that in mind because we're going to talk about that later. A second thing that we did was to develop family genome sequencing. As here you can see, we looked at 41 families that had multiple bipolar affecteds and compared that with 34 normal families. We identified 12 genes that generally fell into the neurotransmitter class whose variants were expressed at very large frequencies in the disease as compared with the normal families. And then what we did was to go to 3,000 people with sporadic bipolar disease, sequence these genes there, and show exactly the same thing was true. So we had a logical rationale to begin thinking about bipolar disease, but the really striking thing is almost all the affecteds had 2, if not 3 of these variant genes. So that changes how we think about certain complex kinds of genetic disease. And the final one, making a bloody window into health and disease with a company called Integrated Diagnostics that we started about 5 years ago. We used mass spectrometry to identify a panel of 13 blood proteins that really had a remarkable ability to distinguish benign nodules from neoplastic nodules in the lung. And when you looked at it carefully, about 40% of the benign nodules could be picked out, and that prevented more than a third of the unnecessary surgeries. Yeah. And that saves the American healthcare system $3.5 billion a year just with that simple diagnostic. So you can begin to see how these tools not only are going to transform how we understand disease, how we treat disease, but how much we have to pay for disease as well. And it was then about 2013 or so that there were 3 really interesting convergences that more fully defined exactly what P4 medicine is for us. And those were systems medicine, big data and its analytics, and parenthetically, the digital revolution that's coming with devices as well. And finally, consumer-driven social networks, and I'll explain that latter one in just a moment. Now, the first 3 Ps, predictive, preventive, personalized, are pretty straightforward, but the participatory, the essence of that P, is it means that the individual is at the center of their health care and they make the critical decisions, not having some doctor look at big populations and kind of guess what might be good for you. And again, you'll come to understand what that means in just a moment. But let's, in the next slide, contrast P4 medicine with contemporary medicine. And they differ in 6 ways. So one is P4, we could have the next one, is proactive rather than reactive. 2, it's focused on individual patients rather than on populations of patients. And 3, it has a major focus on wellness and disease as well, but contemporary medicine is 98% focused on disease. And of course, it's about generating these personalized data clouds that I talked about that are dynamic, that will collect across the rest of your life the data that will allow you to assess both genetic and environmental contributions to your well-being, to your health. Fifth, there is this really interesting idea that the way we do clinical trials now is completely broken because you take 30,000 patients and give them a drug or a placebo, you extract the data into curves, and it's all wrong. Every one of those patients is unique genetically and individually, and when you average them, you eliminate the signal and you increase the noise. And the drug companies have proven this time and time again with billion-dollar ventures. So what P4 medicine does is to assess each individual from their data cloud uniquely. And when we have large enough collections of individuals, We can stratify them according to the properties we're interested in, response to a given drug or so, but the stratification will be based on what they are uniquely, individually, rather than smeared population averages. And that's really a critical point. In the future, N-of-1 experiments are really going to be critically important, not for statistically proving something, but for showing us where we need to go with the proper kind of studies in the future. And of course, the final point I made was this idea of patient-activated social networks, and they're key for three reasons. One, it is a superb way to teach patients and consumers about the new P4 medicine. Two, we've demonstrated in a very small scale that patients can get together in these networks and do crowdsourcing and learn how to improve their health in ways physicians would never have thought of. And three, these consumer-activated social networks, I think, are really going to be key for advocacy in the future, forcing an incredibly conservative healthcare system to change. We're here today exactly where we were with the Genome Project in 1985 or so. And the same parallels are being played out in many different ways. So this, on the next slide, P4 medicine is really just about two things. One is quantifying wellness, the other is demystifying disease. And of course, what we know today is 98% of society's resources go into disease, and 1 or 2% at most go into wellness. And in fact, wellness has a very bad reputation because there are a lot of questionable practitioners, and it's never been studied scientifically before. What we need to be able to do with wellness is create metrics that objectively and quantitatively can allow you for each individual to assess their degree of wellness. And I'm going to tell you exactly how we're going to do that in just a few moments. What I would predict on the next slide is, we need to stay with that, is that we're going to split off, starting about now, a brand new industry called the wellness industry that will be distinct and different from the healthcare or disease industry. And my prediction is, in a 10 to 15 year period, the market cap of the wellness industry will far exceed that of the healthcare industry. And the final point that I would make is wellness is really going to be critical both for the wellness industry, in the sense you need the metrics to assess how healthy we are so we can optimize our own individual wellness. But wellness is really critical for the disease industry, because the only way we can effectively deal with disease is to start with well people and watch them transition into disease and capture the disease at the moment of transition, where we can use these systems approaches to understand the earliest mechanisms of disease, create early diagnostics and drugs, and then flip people back to wellness very early on. So you'll come to understand in more detail about that. Just to emphasize how important wellness is going to be, if you extrapolate over the past 10 years the increase in life expectancy, you can make a pretty compelling argument that of the kids born today, more than 50% will live to be 100. And the really critical question is, how are they going to live the last 30 years of their life? Are they going to be physically and mentally capable of doing the proper kinds of things? We just did an absolutely fascinating study of people that are 110 years or older. And the one thing I learned about them that really is interesting is if you get up into the hundreds, when you die really quickly. So the whole objective for wellness is going to let you go right up to that point of your system's crash, being perfectly capable physically and mentally, and then boom, going down. Because one of the arguments I always get, well, half your healthcare dollars are spent in the last year of your life. It won't be true if we go to that point. So this is, I think, really an exciting concept. So let's talk about this first proposal. I wanted to study 100,000. We still do, but that's going to take a while to get the people and to get the resources. But we have, over the past year, done a pilot project on 107 individuals, and that's what I do want to tell you about. Nathan Price is a faculty member at ISB who's been a terrific co-PI on this entire project. So we have kind of 4 major quadrants of data, but you'll see it's a little more complicated later on. We get a complete genome sequence. We get blood, urine, and saliva every 3 months, and from that we get a variety of different studies, which I'll define for you later. We get your gut microbiome every 3 months. And then finally, we do continual self-tracking with Fitbit and blood pressure and pulse and weight and things like that. So the whole idea then is to create for each individual, if we could have the next slide, this dynamic data cloud, and they are what is going to allow us to predict this future of wellness and disease for each of us. And of course, what's going to happen once we've created these dynamic data clouds is we can integrate them and model them for the individual to create these actionable possibilities. And as I said before, an actionable possibility is a specific suggestion that if you adopt it, will either allow you to improve wellness and or avoid or ameliorate disease and so forth. We found that these actionable possibilities can arise from individual data types, but even more strikingly, when you integrate 2 or more data types together, you really get a lot of actionable possibilities, and we'll give you a whole series of examples of what these are all about. Let me emphasize that what is critical to this whole approach are coaches who understand both the biology of what an actionable possibility is and can be articulate in explaining it to the consumer, the individual, but they also have to be psychologists that can put that actionable possibility in the context of what the fundamental health desires of the individual are, so you can persuade them to change their behavior. We had 2 spectacular coaches that could do that. And what we can say is more than 70% of the actionable suggestions we gave to the 107 pioneers were acted upon. And you'll see the results of these studies later on. So again, if you think in a broader context about a cohort that you're looking at in this way, creating these dynamic data clouds that change every 3 months and so forth, you can quickly see that you'll be able to distinguish 2 groups, those that remain well or increase in their health and those that transition into disease. Now, what was interesting about the 107 pioneers is if you lined up a little scale next to each of them going from 1 to 100, where 100 is optimal wellness, I would say this 107 people that all thought they were well probably averaged about 30. Just to give you an idea, you may think you're well, but I'll almost guarantee you there are lots of things you can really do to improve yourself. And we understand now how to get the metrics that will be required to measure in an absolute sense wellness. And in fact, I'm quite sure we'll be able to measure wellness from a psychological point of view as well as from a physiologic point of view. And of course, if we see those that have transitioned into disease, we can follow them back using these data tranches to the point of their divergence from wellness. And once we identify that, we can use these systems approaches, if you could advance it one more, to actually develop, one more click please, a diagnostics and therapeutics that can divert the person at a very early stage from the disease back to the wellness trajectory and saving society all of the downstream money that would be required for a normal disease evolution there. So on the next slide, you can see that the 107 pioneers used, this is from a Nature News and Views article that came out about the time we started this thing. And we had really 7 objectives in carrying out this study. So one is to create a baseline for participants of health. The second was to establish actionable recommendations and so forth. The third was to give the coaches the opportunity to persuade individuals to understand and accept these and embrace these actionable possibilities. We wanted to see whether we could see clearly these wellness to disease transitions. On the next one, the database and analytic infrastructure, and on the next one, create the conceptual framework for establishing the wellness metrics. And let me say, I think we've, in this initial study of 10 months or so, really accomplished all of those. So if you can click through these rapidly, the blood gives us clinical chemistries, metabolomics, a whole series of proteins. Urine is mostly about stress, as is saliva. The stool is about quantifying bacterial species in your gut, and of course, the self-tracking is exactly what it's stated to be in the personal health status on a quarterly basis. So that was an enormous amount of data then to gather from each of these individuals. We went to the literature and got all of the database information on actionable possibilities in as many different data types as we could, and we ended up constructing a graph database that had 300,000 nodes and 2.4 million edges, where the nodes and edges relate actionable possibilities to genetic variants, to all the other kinds of environmental measurements that we can actually make. And on the next slide, you can see what we were really looking for were not one type of transition, but really 4. Many people went from less to greater wellness. Some actually went, at least temporarily, from greater wellness to less wellness. And of course, the same was true. We saw beautiful transitions from wellness to disease, and then transitions back from disease to wellness. And I'll give you a few examples of all of these things, and we'll just give you some stories about the 107 pioneers. So in this case, we are looking just at the initial clinical chemistries that we drew, and you can see that 91% of the well people had nutrient abnormalities that for the most part could be dealt with really easily by supplements, vitamins, but some of them were missing absolutely critical things that were really necessary for their health. The whole question of inflammation was a major one. And this is something doctors don't handle very well. We had a really interesting case of an inflammation that had been around for about a year or so. And it turned out in this woman, and her physician had tried everything he could to figure out what it was. It turned out it was an enormously inflammation-producing special microbe in her gut, microbacteria, and we gave her a probiotic that got rid of it, and it turned the inflammation around really quickly. So you can, cardiovascular abnormalities, diabetic abnormalities, more than half of the people had these various possibilities. 100% of the pioneers had actionable possibilities, and this is a great yuppie possibility. We had three individuals that had extremely high levels of mercury. 2 of them ate excessive amounts of tuna sushi, and what happened when they switched from tuna to salmon is their mercury levels plummeted, and at the end of this thing, their levels were absolutely normal. So don't eat those fish that are high up on the evolutionary ladder of fish. The third one was interesting. She was a person that had old-fashioned amalgam fillings. and she had them all replaced, and the mercury started coming down. So, and we ended up with 3 people that had enormously high levels of lead, and that's a deadly one. That kills neurons. And here with lead, you need to have chelating therapies, and we instituted those, and those all started to change. That takes longer to change and everything. Now let's talk about actionable possibilities from 2 or more integrated data types. And the next slide is one of the most spectacular data slides I've seen in my career. So with that as a teaser, so what this slide is a subset of 35,000 correlations that we've made, statistically significant correlations between a piece of data in one of these 5 major data type groups, okay, and a piece of data in another one. So what do these correlations between different data types tell us? They give us a view of human biology we've never had before. They give us insights into actionable possibilities. They give us insights into networks that are playing key roles that we had no idea were a part of the processes that we're interested in. And I'll give you a really interesting example. So there was a statistically significant correlation between one bug in the microbiome and it was a negative correlation, and bile acid. So what we did was we used web searching to find one paper that said this bug did indeed destroy bile acid really effectively. And you know, in pregnant women, there is a horrible condition of cholestasis that occurs in the 3rd trimester because with all the hormones that are operating in the woman's blood, it blocks up the bile tract, it spills bile acid out, and they get these gruesome sores and loss of appetite. Everything tastes acidic. It's miserable for 2 or 3 months. Our hypothesis is we can create a probiotic with this organism, give it to those women, and it'll manage the bile acid. Now, is that true? We don't know. But the point is, those are the kind of things that we're going to be discovering from just a casual glance at the information we have. And I'm going to show you one more data slide that comes only a little bit behind this at the very end. But this is a glimpse of human biology no one has ever had before. And of course, what's so wonderful about it is we have to figure out what it all means. Okay, let's talk about a wellness to disease transition, hemochromatosis. Next slide. So we found that 2 people from genetics had a defect, had homozygous defects in a gene that causes hemochromatosis, which is high levels of iron that are present in the blood. And it can be a really deadly disease because it attacks the joints, the pancreas, the liver, and finally the heart. And usually it presents with the heart failure, but by then you're a chronic invalid the rest of your life. And so what happened, and this is really with the most spectacular individual here, he had 2 concierge doctors on the West Coast and the East Coast that had been trying for a year and a half to figure out why he was getting increased arthritis that limited his ability to hike in the Cascade Mountains. And when we first got the gene thing here and then measured the iron levels, it was obvious that he had hemochromatosis. And of course, it's enormously actionable in the sense all you have to do is take a unit of blood every month until you get it down to normal. And then every 3 months, you can keep it more or less at a normal level. And by the end of the 10-month study, his arthritis had reversed completely. So again, it goes to show you with this kind of study what you can do in terms of looking at transitions. What was really important is he had genetic testing done on other members of his family, and there were other people that were really at risk, and you take the appropriate actions on that. On the next slide, we'll see the gut microbiome. And what, if we could have the next one, yeah. Here are the 107 individuals, and the different colored bars here indicate different ratios of different bacterial species, and it's kind of a continuous distribution from one end to the other end. Curiously enough, these two people at the extreme end here are computer scientists who are among the best in their class in the US. So I'm trying to make the argument it was their microbiome that determined it. So see, that's a good example of a correlation, but it almost certainly is a totally wrong correlation. So, but what was interesting is, if we could have the next slide, please, that we could bin the 107 individuals according to 91 variants that predispose to Crohn's disease, from very low to medium to high to very high. And then we could, on the next slide, layer on top the microbiome. And what you can see is these two pro-inflammatory bacteria go from about 1% or so up to about 10 or 15% if you total them up all together and everything. And it's a linear correlation between the genetic risk you have for the disease and those microbial species that are present there. And on the next slide, we know that this is a pretty good call because the one diagnosed Crohn's disease person we had fell right here in this high category. So this opens up a lot of really interesting possibilities of using probiotics to deal with Crohn's disease, which I'm sure some of you out there have it. I know, in fact, one has it. And we could talk about that kind of thing in the future. On the next slide, there are now probably close to 70 diseases for which there are genome-wide association study markers, that is GWAS, genetic markers for these diseases. And an example that I'll use is they studied almost 200,000 individuals, half of them for the discovery set, half of them for the validation set, and they identified 72 loci that predispose to increased elevation of cholesterol levels. And on the next slide, what we can do for each of the pioneers is we can take the two types of variants, those that cause decrease in overall cholesterol, those that cause an increase in overall cholesterol, and we can use the probabilities associated with these to determine genetic risk profiles for each of the individuals. And of course, when we do that, on the next slide, you can see some really startling things. So this is one of our larger families, 10 individuals where the high risk is on the left-hand side, the low risk is on the right-hand side. The profile here is for HDL, the good cholesterol, and of course, high risk has low levels of it. Whereas the LDL, the bad cholesterol, high risk, you have high levels of it, and the low risk have very low levels of it. So a couple of things are important. One is the correlation between the genetic predisposition and the phenotype is really striking. And that's really true, especially with LDL. Here, it's very, very good, except for two really interesting, if we could have the next one, outliers. One that's way above his genetic level is performed to get HDL. And he's a triathlete, and he was number 2 out of 107 in terms of wellness at the beginning and at the end of the study. So you can, with proper environmental lifestyle changes, do very well with HDL. But if you go over here, you can't even tell which one he is. This one is much more highly genetically controlled, and in this case, you're probably going to have to use, you're probably going to have to use for the high-risk people drugs like statins. Now, what's really interesting is this person who performed way below their level, and the reason on the next click that's interesting is the level here is the same as the level here, And if you were a physician, you'd treat all 3 of those people the same way, and yet only 2 should be treated the same way. The high risk should be almost certainly given statins. This person should have a change in lifestyle. So by putting together 2 different types of data, you make the proper call for how to optimize wellness. And we have at ISB actually now almost 7,000 genomes. I haven't updated this now. What we can do with these 7,000 more or less normal genomes is we can create the same kind of genetic models in the normal ones, and then we can map the individual genomes onto those higher level on the next click level. So here is what the 2,000 genomes we did first looked like, a bell-shaped curve for risk or increasing risk for LDL as opposed to the number of individuals. And what we can do then is begin mapping to say where you are in this risk level with regard to the population. And on the next slide, what is really striking is with GWAS data, look at all the different levels of disease that we can study. And as I said, there are probably a number of, probably another 20 or so, if we could have the next thing, that are there. And this gives us an enormous ability to make calls about genetic predisposition. So what we're doing for each of the pioneers is we're going to tally all those scores up. Where they aren't actionable, we'll keep them until they become actionable. Then we'll inform the individual to go immediately to their physician and get in line for the treatment. And I'll show you an example of where we're going to do that in just a moment. So it's an enormous incentive for coming into the study because you're going to be followed at an incredibly detailed genetic level, yet you're not going to be worried about things you can't do anything about. For people who are infinitely curious, we'll give you all the data. You can have it if you want. But most people, I think, would prefer to be told when something can be done about it. So on the next slide are profiles for Crohn's disease, for obesity, for late onset Alzheimer's. Really interesting double shoulder. I have no idea what that means. And then for psoriasis with this very long tail. We have one of my colleagues who's had psoriasis all his life, who's way up here at the highest end of the level, and he kind of confirms the power of the statistical analyses here. So a study we're going to do is a longitudinal high-dimensional data approach to Alzheimer's disease. So on the next click, there are 3 sets of markers we can use to determine the risk of a given individual for Alzheimer's disease. And we've done that for the ApoE4 allele, and what we found are 22 individuals are heterozygous for this allele, 2 are homozygous for the allele, and these are serious genetic markers. If you're homozygous, you have a 65% chance of having Alzheimer's disease by the time you're 65 years old. If you're heterozygous, one bad copy, you have about a 12% chance of getting it. So you really want to be followed. So we've mapped then individuals against the 2,000 normal genomes to determine risks. And on the next slide, what we're going to do is we're going to take 200 high-risk Alzheimer's individuals, obviously 20 of those from this study, but we're getting them from other studies as well, to do these high-dimensional data-rich analyses to look at the earliest transitions and come to understand the disease-perturbed networks and the early blood diagnostics and early therapeutic targets. And the really key point on the next slide is a collaborator of ours at UCLA is convinced that if we can determine the earliest transitions, one can use therapy, exercise, supplements, diet to actually delay the onset of disease for 10 to 15 years. And if we can do that, we might even get to the point where we'd have a cure. So all of these people will be accessible for this kind of approach. So you can see what you can do with this kind of study. And finally, I'm just going to very, very quickly show you how we're going to get metrics for wellness. So the metabolomics, just to remind you, we're looking at 700 different metabolites in the blood. And what we've done here is we've created a measure of activity for the various 100 pioneers as identified by, or of weight change, as identified by BMI. And what we can show in kind of a heat map, and I'm not going to explain all the details, is on the next click, there are a set of metabolites that increase as the BMI decreases, and that's good. Those are the metrics you want for wellness. And conversely, on the next click, you can see that there are a set of metrics that increase as the BMI increases, and of course that's a negative metric for what you have. And there are really interesting ways you can play these things together. On the next slide, what we're going to be able to do is to take all of those metabolites and map them into their cognate networks and come to understand in considerably more detail all of the things that are involved in obesity, weight gain, metabolic syndrome, and all of those kinds of things. It's going to allow us, in a sense, to look into the dark matter of human biology in ways we've just never been able to do it before. So here's the final really spectacular slide, and it's absolutely a fascinating slide. If we have your complete genome sequence, what we can demonstrate is all of us are double knockouts for a handful of genes. So the really interesting question is, and let me just say, most of the genes that we've seen that are double knockouts in these people are genes that don't have activities ascribed to them at this point in time. So what we can do is correlate with the double knockouts all of the metabolites, all of the proteins, all of the clinical chemistries, all of the gut microbiome, and we can begin to start understanding where is the network hierarchy into which these genes begin to fit. Again, it's a chance to look into the dark matter of human biology we've never been able to look at before because we've done these global, integrative, dynamical analyses. On the next slide, go ahead. You can see then that we have 35,000 of these statistically significant correlations. And on the next slide, we actually have looked now at 5 or 6 different diseases, plotting genetic risk against phenotype, and in every single case, the correlations are absolutely beautiful. So they reaffirm the interrelationship of phenotype and genotype and the validity of making these genetic risk calculations. And finally, on the next click, you can see how we can use heat maps in a way I haven't really described well to actually identify biomarkers that are going to be key metrics for wellness, but they're going to let us probe those dark areas of human biology that we don't have any understanding of. All 107 pioneers had multiple actionable traits. And of course, since when the environment changes, you get new actionable traits, following the dynamics is absolutely critical. And on the next slide, the insights were really, first one please, your genome doesn't control your destiny, it controls your potential. And for most genetic things, you can, through appropriate behavior, eliminate risk and difficulty. Not all, but many. Number 2, I think the most revelatory thing was that almost all the pioneers felt for the first time they could take control of their own health. They had an understanding of what kind of knowledge was required and how to make decisions, and the coaches were enormously effective in teaching this to people. And as, next slide, a mute testimony to how effective the study is done is almost all the pioneers want to move into the next study, which we'll talk about in just a moment. And finally, 70% of the actionable suggestions were adopted, and it's remarkable testimony to the job that the coaches have done. So, on the next slide. So, what have we gotten out of this project? Well, one, we've got a catalog of about 150 actionable possibilities. Two, we've learned to optimize wellness and avoid disease with these actionable possibilities. Three, we're beginning to get the metrics for wellness. Four, we're beginning to see early disease mechanism transitions, and we're going to be taking on populations that are at risk for a wide variety of different diseases to look again at these earliest transition points. We're driving the development of old and new assays and analytics. And for example, one thing that we don't have here very effectively is the immune system and immune assays. And we have great ideas on how to do that. It just costs a little bit more money than we can afford at this point in time. But the ideal, of course, in 10 years, what I hope is you'll have an app on your cell phone that can take a drop of your blood and give us 95% of the measurements we need to make the calls that we're talking about here. And that requires some new types of chemistry, but I think we can get there. We have a database of wellness and disease transitions that actually is going to be really transformational, because from this, we're going to catalyze the emergence of this new wellness industry and create a whole series of companies that will be focused on different aspects of wellness and so forth. And finally, this really has brought P4 medicine to the healthcare system, improving quality, decreasing cost, and promoting innovation. So, where are we going to go from here? We've done 107. This next 18-month period, we're going to do 10,000. We're going to increase it 100-fold, and I'll tell you how we're going to do that in just a second. And then after that, 50-fold and up to 50K, and then finally up to 100K. On the next slide, we're going to take this project in two directions. One, it's going to be academic. Here, we're going to do discovery. We're developing technologies. We're developing analytics. And we're really going to focus on both wellness and early disease transitions with all sorts of partners. And we actually have lined up probably 30 different partners, including 3 different countries. So one country we'll probably be doing this with is Iceland, and a second, interestingly enough, is Indonesia. I can talk about those if people are interested. The second objective is we're creating a company that's going to reach out to consumers, wellness, science, and it is this company that's really going to push over the next 18 months the creation of 10,000 data clouds. And the relationship between wellness science and ISB is that ISB will do the analytic work on the 10,000 data clouds, and of course, wellness science will have first rights of refusal. But we're pushing this because we can get society to pay for the 10,000 deeply phenotyped data clouds, whereas the Pioneer project that we did initially, I had philanthropy pay for that, because when I went to Francis Collins 6 months ago to ask him about whether he'd consider funding this project, he said basically no way would he consider funding it, because the NIH had so many cutbacks. And I don't know what he would say now that this precision medicine has been announced, but what precision medicine is not very precise, I'll say that. Okay, on the next slide, my vision is, one, I think P4 medicine is going to be the center of the healthcare paradigm in the future. I think health is about assessing genetics and the environment in these dynamical data clouds and giving you accessibility to actionable possibilities. It's really going to be the key way to do it. The two thrusts of P4 medicine will be quantifying wellness, demystifying disease, and of course, wellness is going to allow individuals to reach their full health potential, and it's going to allow us to study the wellness to disease transition. It's going to revolutionize disease. I mean, if you think about it, it's really ironic. How do we study disease today? We study it when you get sick and you go to the doctor and he tries to figure out what to do. It's too late. That isn't what you want to do. So, and on the next click, so we think a wellness industry will emerge, and there are going to be enormous economic opportunities for people who participate in this revolution. And here are 5 reasons why I think the cost of healthcare will come down. By digitalization of medicine, I mean the digitalization of the assays we need to make the data clouds. And I gave you the example of the human genome being $100 in 5 years or so. And I think all of the clinical chemistries, but all the rest of them will go through the same kind of change. A systems approach to disease, that is early diagnosis and identification of therapeutic targets. The lung cancer one, where we can save society $3.5 billion a year, is a nice example. The dynamic data clouds to optimize wellness and minimize disease are going to save an enormous amount of money, as does the reversal back to wellness. And of course, I think individuals taking responsibility for their own health is going to make an enormous difference too. But when you go to insurance companies and say, look, this big revolution is coming, they're very cynical about it, and they say, prove it to me. So we will be proving it to you, and I think you can prove it to you with the consumer approach, where the consumer is going to be paying for the transformation of healthcare rather than government. Here are the kinds of skills that were necessary to carry this out. Again, Nathan Price has really been a full colleague in all of these kinds of things. And then finally, on the next slide, let me make a couple final comments for students and perhaps faculty, and then we'll close. So on the next slide, so we can click through these. You need a passion for your science, and you need to have fun. I think that's the most important thing in science. Don't do it if it isn't fun. Go to Wall Street if you enjoy making money and make money. But I don't think you'll find that very fun at all. But on the next slide, have confidence in your own ideas and visions. I mean, as I think back through my career, every single one of those 5 paradigm changes I talked about, there was enormous skepticism. I would say when I started the Institute for Systems Biology, I'd say 90% of the biologists were skeptical about whether systems biology was anything more than simple hype. And what's really ironic is one of my good friends from MIT felt that way really strongly in 2000, and he was the chief author of a report from the National Academy 10 years later that said systems biology is the future of biology. So people change. You can always hope for that. On the next slide, it's really key to work with good people because they push you to be what you can be. And so wherever you have a choice, go work with terrific people. And choose significant problems. I mean, my PhD thesis advisor always used to say, there are authors and there are librarians. You don't want to be a librarian putting the books away. You want to be an author writing the books. So, choose big, significant problems. Thinking outside of the box, I'm always amazed at how few people can really think outside the box. And I think for the faculty here, there are really terrific ways to teach people to think outside the box, but you have to do things very differently than you traditionally do them, and we can talk about that if there's time. I think determined optimism is utterly essential for science these days because, look, it's hard in a lot of ways, but, I'm utterly convinced that determined optimism makes things happen the way you want to. You can inspire, you can You can push, pull people, and it's really critical because somebody has to say in the midst of Chicken Little and the world is falling down that, oh no, these challenges really are opportunities. You're just not looking at them in the right way. Cross-disciplinary training is really, really important. As a biologist, you should learn several other disciplines. And in fact, my argument would be, I think every biologist should be an equal dual major where you pick biology and something orthologous to biology, computer science, mathematics, applied physics, whatever, and you really learn that well. And you know, one of the big arguments is, oh no, we don't have enough time to do that because we just have to keep very much time to teach the way we teach now. And that's utterly wrong, because if I think back even about my Caltech graduation, I would say 60% of my classes were vanity classes where the professor taught you in enormous detail everything they knew about their own little micro-topic or in related ways, that kind of thing. And I think I had two really spectacular teachers who did it the right way. So I had Richard Feynman for freshman physics, and I had Lyman Colley for freshman chemistry. And they were conceptual thinkers that never got bogged down in the detail. And that's, if we do that, we can really simplify the list. So, and I think mini courses are really a terrific way to broaden your knowledge base and everything. You need the computational training to deal with big data. Look, big data, there's even big data in history now. There is a really thriving branch of history that's putting into the computer everything we know of history, and they can ask questions historians would never, ever begin to ask. So whatever you do, you're going to need to be able to deal with data, and you're going to need to be able to think about noise, because big data has lots of noise. Learning to work together in teams is really, really critical. A lot of the problems we take on are really hard, and a single person can't do it by themselves. And on the next one, learning is lifelong. That's kind of self-evident. But my final point on the next one is I think I really recommend highly that we change careers every 10 years. So if I can have the last slide. And this is something that Norton Simon taught me early in my career at Caltech. And he said, life has a natural curve, and you go up, and most people have a bell-shaped thing, they fade away. But if in fact you go up and somewhere in here, you decide to change careers, you become insecure, you open up your ability to learn, you get excited about things again. And in fact, if you do that again and again, you don't ever have to go down, as long as you're in good physical shape. So, thank you.